And my question was basically when you're talking about certain immune cells, and you talked about the downstream effect. Some of them have a downstream effect. Can you go into a little bit more detail what that means by downstream effect?
So, T cells play a huge variety of roles, and I'll be perfectly honest, I'm a lot newer to this side of it than I am to some of the others. But basically, there's a lot of things that they can do in terms of attracting other cells in that will help create an environment that will help to basically destroy those tumor cells. They may cause inflammation. You can get kind of chemical gradients that get set up in the body based on things that are released from them, etc. Some of them may kind of go into storage, I guess you could call it, and come back into play later. There's a lot of different things they do.
Thank you. Erin, did you have anything you want to add to that?
Yeah, I was just gonna say, so there's also sort of going back to what Michelle had said earlier, there is a subset of T cells that are sort of the warriors. And that's actually what's in this diagram here, the CD8. Those are the ones that will actually go and kill the cancer cells. And then, and I think we all sort of know we get vaccinated. I hope we all get vaccinated. Um, and so we know that our body has a way to remember that and make those antibodies when we get reinfected. And those are actually B cells. That's a different subtype of immune cell that makes those antibodies. But you also have T cells that remember things that they've fought before. And those sort of nicely are called memory T cells. And so there's a whole range, like what Oli was saying, of different kinds of T cells that serve different functions.
Thank you.
All right. So once we found these family interacting proteins that are, um, showing that there's a decrease in interferon gamma production, which is basically just what's coming off those T cells, we wanted to see if that was because we just had less of the cells, right? Or something of that nature. So we went ahead and looked at how many cells, how rapidly those cells were multiplying. And so we didn't really see any differences, right? We tracked this over the course of several days. And even if we didn't have the FIP2 there or the FIP3, proliferation didn't change, whether we looked at colony number or area.
Could
So
I
that,
stop you right there? A
yeah,
lot of times there's terminology that maybe newer advocates or even those who've been around for a while might not understand. It talks about proliferation, and you mentioned that term. Could you explain what proliferation is?
yes, of course. Sorry. Proliferation is just basically multiplication, expansion of these cells. So that's just looking at how much the cells grew in this context. Yeah.